Thank you all for coming for this meeting, and thanks to biocrates and Vijon and Manoj for giving this opportunity. So I know that we have a very small time, like maybe 15-20 minutes to go through a lot of stuff. I'll try to be brief. I will touch upon two stories today. One is on drug mechanism of action. Another one is host-pathogen interaction, both in the context of malaria. So I just want to start with one introductory side. I won't say too much about these parasites. My lab is actually focused on infectious disease work, and we deal with pathogens such as malaria parasite and other related parasites like toxoplasma and a few other parasites, including in animal infections. But today we'll just talk about Plasmodium. And of course, these parasites, they infect humans and they cause really bad diseases, malaria and toxoplasmosis. There are drugs to treat these diseases, but as most of you would be aware, there is always this problem of drug resistance and ineffectiveness of drug. And all over the world now, WHO is going for malaria elimination programs. So we need better and better drugs, as well as we also need to keep track of whether the parasites are becoming resistant to newer drugs, which we are discovering and things like that. So I'll tell you one story about how we did find some novel molecules which are acting on already established pathways and how that can affect discovering new drugs. So I just want to sort of describe this, oxidative phosphorylation, which is in the mitochondrial inner membrane. And most of you those of you who have studied biochemistry will remember this. So this is a typical oxphos. Only the fact is that this representation is from the malaria parasite. So it's not the same as humans. There are some differences. Like, for example, if you look at the complex 1, this oxphos enzymes are arranged in various complexes. So if you look at complex 1, that is completely missing in malaria parasites. But there is complex 2, complex 3, 4, and 5. Okay. Now, complex 3 is the key complex because it couples the electron transfer from other dehydrogenases to complex 4. And this coupling is very important because unlike in humans, since these are missing complex 1, the only way to pump protons across the membrane is through complex 3 and complex 4. And that proton gradient is important for ATP synthesis, which is the complex 5. Just give me a second. Let me see if I can get my pointer. Yeah. This mitochondrial respiratory chain is a well-known drug target, and there are many chemicals which people have used as experimental inhibitors, as well as there are some like atoque-1 which are even used as drugs. The problem is, for example, with atoque-1, it targets the cytochrome B, okay, in this complex 3. And cytochrome B can become mutated and it can become resistant to atoque-1. Okay, so even though atoque-1 is a very powerful drug, its IC50 values in single-digit nanomolar values, and but still resistance is a problem. So one of the things is, but the target is a very good target, and so if you can identify more molecules which act on the same target, which is the complex 3, then you can easily kill the malaria parasite. So we developed. So I'll just skip this because it's all introduction. So we developed a mass spectrometry based assay to look at the biochemical effect of inhibiting the respiratory chain with atoque-1. So what you see here is, what I'm showing is a simplified version of the pathway for the synthesis of uracil monophosphate, starting with glutamine. And there is this enzyme called dihydroorotate dehydrogenase, which is dependent on complex 3, which is the cytochrome BC1 complex, for recycling of electrons through the Q coenzyme Q. And if cytochrome B is inhibited by atoque-1, dihydroorotate dehydrogenase will not function. So it will stop. As a result of which, we will get accumulation of this substrate for the enzyme called dihydroorotate. And because the flux is very high, this really accumulates very fast. So you can see below very quickly, I'm going to show this. In untreated cells, you hardly see accumulation of dihydroorotate, but in atoquan treated cells, you can see a huge increase in the accumulation of this. Of course, downstream, if you look at UMP, there is a decrease in UMP, but because UMP pool is very large, the turnover is very slow. So these experiments are done just after 1 or 2 hours of drug exposure, in which time UMP cannot be fully depleted. Okay. So now we have this assay, and if you look at parasites treated with atoquan, you will see here that, in the presence of atoquan, you will see that DHODH is, accumulating. So we were looking at many molecules, which we, based on the chemistry of the molecules, the structural similarity of the inhibitors, we suspected that they will act on the cytochrome B. And so we screened some of these molecules. We got all the IC50 values. I won't go through this. It's just a routine antimalarial screening. And for some of them, we saw, at least I'm showing here 3 examples. So you can see dihydroorotate accumulating with atoquan, which is the first bar. And then you can see 3 other molecules. They're all giving increased, dihydroorotate. So which means they're acting through the same pathway. So like this, we can use mass spectrometry for the discovery of new molecules acting through known pathways, creating the, targeting the same biochemical mechanism in the parasite, creating the same kind of metabolic block, and etc., etc. So this was exciting. So then what we proceeded to do was to look at mitochondrial intactness by looking at this JC1 dye, which if it is a red color, then the mitochondria is doing fine. If there is no red color, the mitochondria is not doing fine. So you can see with atoquan in wild type, normal parasites, there is no red color, which means the mitochondria has collapsed. If you look at the molecule, one of the molecules we found, LW16, that also collapses the mitochondria. So these are just confirmatory experiments. So then we proceeded to generate mutant parasites which don't respond to atoquan. So you can see the inhibition assay here that the wild type is shown in red, but you can see the mutant is at least 2 orders of magnitude less sensitive to atoquan. And you can see below here the data, like atoquan is single-digit nanomolar, whereas this mutant is like 20 micromolar. So it's almost more than 1,000-fold resistance is there. So we characterized this resistant parasites in many different ways to prove that the mutations in this parasite affect only atoquan activity and not other antimalarial activity, which is shown here. But I'll just go ahead. But the interesting thing is now we want to see what is the mutation which creates resistance to atoquan in the lab. So we did genome sequencing, we identified the mutations, and very interestingly, the target cytochrome B was not mutated, which is what we expected. But another enzyme called dihydroorotate dehydrogenase, which I just introduced to you in my earlier slides, that was mutated. So now what is happening is a mutation in DHODH is decoupling the pyrimidine biosynthesis from the electron transport mechanism. So how it is happening, we still don't know. And interestingly, similar mutations have been seen in the clinical isolates as well. So people have sequenced malaria patients, malaria parasites isolated from patients to show that mutations in DHODH can cause atoquan resistance. So we are seeing it here. So what I just want to show, this data is very important here. This is one of our new molecules, which is the LW16 molecule, which acts through the respiratory chain in the mitochondria on the parasite, right? Then we show from this experiment that even the mutant parasite, which becomes resistant to atoquan over 1,000-fold, is still sensitive to LW16, which means although the target is the same, the pathway is the same, somehow LW16 whether it is binding to cytochrome D or its interaction with DHODH. What is different, we still don't know the full mechanism, but we know for sure now from this data that the parasites are still sensitive. The resistant parasites which don't respond to atoquan are still sensitive to LW16. So this got us excited because we now have like a new antimalarial compound which acts through a very well-established target pathway. So we characterize this further. So if you look at the mass spec, you can see that LW16 still results in accumulation of DHODH, whereas atoquan, in wild type, it accumulates very high. You can see in the other graph, you can see that atoquan accumulates in the wild type. But you can see here, the atoquan is not as effective on the mutant parasites, but LW16 is still working. So, and we show this again phenotypically that the mitochondria is affected. It's not affected by atoquan because of the resistance, but LW16 is still killing the red color, which means mitochondria is still affected. So like this, so what I just wanted to impress upon you is we can do these kind of metabolomic studies to understand molecular details of how drugs work inside the cells. The next story I just want to jump quickly is this thing. So this is unrelated to drugs, but here we are looking at host parasite interaction, how a pathogen can influence metabolic changes in the host. And it can happen at several levels. It can happen at an immune level, or it can happen at a metabolic level, and so on and so forth. So when you look at plasma metabolomics in the context of infection, we can learn a lot about how the host actually responds to infection and what are the specific changes which are parasite specific or disease outcome specific. So those kind of things we can see. So this is our experimental strategy. So, okay, I just have to say that this experiment we did with animal model of malaria. So this is a rodent malaria model, Plasmodium berghei, which is a very, it's a very virulent malaria parasite. It kills the mice. There are also other malaria parasites which do not kill mice, but this particular type of parasite, Plasmodium berghei, will kill the mice. So our goal was to infect these animals with this parasite, which is going to cause fatal malaria, and look at, as the disease progresses in the animal, what are the metabolic changes that happen in the animal, and to see whether any of the metabolite marker will correlate with the disease severity. So that was one of the goals. And also we wanted to find out how the response from the host happens at a metabolic level and how it might affect parasite division and growth. Because see, the parasites, when they're living inside the host animal, they depend on metabolites from the host for nutrition as well. Like, they scavenge many intermediates, and I'll show you some of that data. So we wanted to see whether the host can challenge the parasite by modulating levels of these nutrients required for the parasite and all this. So this is the experimental strategy where we take mice and we infect the mice at a different date, so that at the time of harvesting, we could take blood from all the mice together on the same day, because we know that in animals, age of the animal also affects the plasma metabolite profile. So we wanted to make sure the animal are all sampled on the same day so that they are all age matched. So we did it like that. And you can see below that as the infection is progressing from towards day 9, because almost by day 10, these parasites are going to die. So day 9 is like they're really sick. So you can see body weight dramatically drops. You can see we did a lot of organ phenotyping also. I'm just showing you one organ here, spleen, which is on day 6, when parasitemia is very high, the spleen is full of infected cells. And then basically the spleen shrinks because there is a lot of cell death and autophagy after that. And this is all well documented in literature. It's not that this is the first time we are showing all this. So then we take, so as you can see from this figure, we just take the plasma from all these animals, and we call them as day 3, day 6, and day 9, or uninfected plasma controls. And then we look at it, you can see that the day 3 plasma metabolites, so these are global metabolites. This is untargeted. We did complete full scan of using orbitrap mass spec. And you can see here, there is very high correlation between day 3 and control. But as you go towards day 9, where the infection is progressing, then you see the correlation decreases, because there is too many metabolites which are showing changes as disease is progressing. So at this point, I would like to say that many of these are just mass features. Some of them can be fragments. So this is just total global stuff. So we looked at few of them, like we wanted to see p-value correlations, and then we could identify those metabolites which really change with good p-values. And so we could focus on that and see what these metabolites are. And if you look at the global data here, what I'm showing on the left side is it's all the metabolites which are increasing, and on the right side is all the metabolites which are decreasing. And you can see as you go from day 3 to day 6 to day 9, the amount of metabolites that are increasing are also larger in number for day 9. Same way for decreasing also, you see many more metabolites are decreasing at day 9. And the surprising thing, which for me at least, is that we see very little overlap between metabolites which started to decrease or increase in day 3, and then as we progress, a different set of metabolites are showing. So and this is very apparent for both increase and decrease. So in this data, we are just showing you only the data for metabolites which are correlating with very significant p-values, and at least a 2-fold change. And all the ones that are shown in red are increasing, the ones shown in blue are decreasing. So these are just mass features. We don't know what these metabolites are. Of course, we can capture some of these metabolites and do MRM, and we can find out what these are by fragmentation, but we haven't done those kind of things right now. And in future, we plan to do that. And you can see very well as you go to day 9, as disease is progressing, some metabolites just keep accumulating, and some others just keep decreasing, depleting. So these are very interesting for us to see whether these actually have any consequence on the disease outcome when the parasites are in the growing rapidly inside the animal. We also looked at certain metabolites for which we had in-house library of standards, and we could identify them and all. And you can see, because our library was very small, I think we had less than 100, 60, 70 metabolites in our library, and we could look at them. But some of the things stand out, like for example, all glycolytic intermediates appear to be decreased in animals when they are progressing towards. So you can imagine that energy metabolism and carbon metabolism is skewed in animals when there is. The interesting thing that came out was this tryptophan metabolism, tryptophan and its metabolites. See, tryptophan is a very important amino acid, because from tryptophan, so many other molecules are derived in an animal body, and many of these are neuromodulators and signaling molecules. I have just put up a few of these. For some reason, as infection is progressing, the tryptophan levels are reduced, and as well as some of these are even immunosuppressive molecules, like 5-methoxytryptophan is a very well-known immunosuppressant, and you can see up to day 6 it's very steady, but then day 9 it just totally drops. So it's like at the immunomodulator level also there is a lot of significant change in these animals. Okay. So the last, I think this is the last data slide. So I just want to say that all these nucleotide precursors like inosine and hypoxanthine and xanthine, these are scavenged by parasites to make their own nucleotides, because the parasites don't have a pathway for purine biosynthesis. They pick up all these purine precursors and then they make their own purine nucleotides. And you can see, especially xanthine, because xanthine is one of the most abundant purine precursor, and it is easily converted to ATP and GTP in the parasite. So basically xanthine is heavily scavenged by the parasites. And xanthine also is part of a pathway which results in the formation of uric acid. Okay, when xanthine is degraded, you get a lot of uric acid. And this uric acid can be toxic to the parasite, toxic to the host. And this conversion of uric acid to allantoin by uricase enzyme, this happens only in mice and some other lower mammals. So this uricase enzyme is absent in humans. And it is very well known that in severe malaria cases, uric acid accumulation in human blood is a very important clinical consequence. And this is one mechanism we see mice are able to detoxify uric acid using this enzyme, but humans can't do it. Despite this detoxification, of course, mice become susceptible to malaria. I don't think this is the only mechanism by which you can control malaria, but this is something the animals are attempting to do. They are increasing their formation of allantoin and then converting the accumulating uric acid into allantoin, which humans can't do. So this kind of very detailed analysis of how an infection actually affects the plasma metabolite levels and what is the host pathogen interaction can be easily studied with the help of mass spectrometry. So I would like to stop here, and I would like to acknowledge and first of all, thank you all for your patience, and then acknowledge a few of my students, Anurag and Tejasri, who have now graduated and who are out of the lab. But these are all work which they had done while they were still in the lab as students. And my collaborators, Dr. Amit Sharma and Dr. Jyoti Chibbar, both of them from IGIB New Delhi, and then Krishnapal Karmodia from ISER Pune. I'm sorry, there is a typo. Amit Sharma and Jyoti are not from IGB, they are from ICGB. So I think this came as a typo. And then I would like to thank NCL and CSIR-DBT for facilities and funding. Thank you all, and if there are any questions, I can take.